As was mentioned, we've been interested in modeling of biological signaling systems for quite some time and have been fortunate enough to be able to interact with some really excellent microbiologists in this effort. And so what I'm presenting to you today really is the outgrowth of interaction with these colleagues and learning all of the unique constraints that exist in their systems. So our microbiology colleagues are very interested in understanding how microbes interact with each other. There is this cell-to-cell interaction. We'll talk about this with respect to quorum sensing. There's also a multi-species interaction, and we have begun modeling of these kinds of systems as well. And certainly as engineers, one of the questions we always ask ourselves is, why are bacteria engaging in these interactions? A joke that I have with one of my colleagues is whether Mother Nature is optimal or not, whether she tries to optimize systems. He believes not. I, as an engineer, am hopeful that she does. But it is clear that there are objective functions, sometimes hard to quantify, that these microbial communities are interested in. How to minimize their metabolic burden, how to increase their resiliency and robustness, and certainly how to engage in community actions that benefit the health of the community. So what we've been endeavoring to do is to try to map these biological objective functions, if you will, or sometimes unquantifiable measures into something that's measurable in order to do design and understanding of these microbial systems. So we, of course, are looking at metrics like delay minimization, fidelity, data rate, et cetera. So in particular for today, we are actually going to have systems in which the transmitters that we're used to and the receivers that we're used to are actually colonies of bacteria. And this is motivated by the experimental systems and the questions that our colleagues are trying to answer from the perspective of microbiology. So we're going to have a so-called transmitter colony and receiver colony, and we want to understand the behaviors of these two colonies. Our motivating example or experiment, if you will, is this MISTIC system that was developed out of Ophelia Venturelli's group at the University of Wisconsin. And this is a system in which they have a fair amount of control in terms of being able to inject certain kinds of food and nutrients, if you will, to the transmitter colony, but they also have control over the propagation channel. And that is to say, they can change the distance. And they were very interested in trying to understand how this distance plays a role in terms of the behaviors that are engaged in with the receiver colony. And given the nature of these microbial systems, there's this nutrient, if you will, that is put into the system, a so-called input signal. The transmitter colony does a conversion of this, and then it propagates through the channel where it's received by the receiver colony, usually through some kind of binding operation. And so it's this conversion of one molecule into another, if you will, that motivated this scheduling work that we're doing right now. And it's important to understand that we have all sorts of biological activities that are happening both within the transmitter and the receiver colony, but as many of us are used to in our studies of molecular communication, we still have diffusion governing the transmission ultimately of the type B output signal to the receiver colony at the end. So what are the goals and the assumptions that we have in this problem that we're trying to frame right now? And I want to underscore that the heavy lifting has been done by my excellent PhD student, John Gursoy, who's actually going to be graduating this spring. So our underlying assumptions is that these biological systems seek to be energy efficient. It is not in their best interest to waste energy. We also have an important assumption, which we are actually seeing evidence in experimental systems, that the faster processing of these input molecules is actually more expensive. So there's a price that we pay with respect to the processing of the signals. And we have some control over this processing. And so what we'd like to understand is what is the most energy efficient way in order for that transmitter colony to process its input molecules before they are transmitted over this propagation channel to the receiver colony. And this has relevance both for natural biological systems as well as synthetic systems. And the experimental systems that a number of our collaborators engage in are actually these purely synthetic microfluidic devices that they've created, where they have a lot of genetic control over those transmitter and receiver colonies. So our new framework here, which we believe is different and unique, is to model each of these colonies, and these transmitters in particular, as so-called schedulers. And so they have control over their processing rates in order to be most energy efficient. On the other hand, there is a desire to minimize the end-to-end delay. And we did realize that this very engineering metric of minimizing end-to-end delay was actually something that was of strong interest to the microbiologists, because they want to figure out how quickly they can induce changes in the receiver colony. So what we're going to see is that we're going to have to balance this input rate of molecules into the propagation channel and try to optimize our use of this end-to-end energy while achieving delay constraints. And our model is going to capture both the diffusive delay and this notion of molecule degradation. So before I get into the details of the model, let's make some contrasts between what we typically do in, say, wireless communications, where scheduling packets is a very common activity. And typically we try to be energy and time efficient, just as we're trying to be in this biological problem. And there's a whole history of packet scheduling works that's been persistently studied. We've been focusing on our own prior work, which was inspired by work out of MIT and Stanford in terms of this molecule scheduling. And the key is that you want to make sure you make a delay constraint at the receiver. You don't want to be late. Now, we would like to leverage this concept for molecule scheduling, but of course, now we have to take our biological constraints into consideration. And what we're going to find is that we have constraints both on being too late, but also being too early. And it's this too early that is a new proposal here. And so let's step back for a moment, although I know a number of you in the community are already aware and actively working on quorum sensing mechanisms, but many bacterial communities and strains engage in quorum sensing. Essentially, they produce or synthesize important molecules, which are then released into the environment. They're called autoinducers. They can also recognize or sense the presence of these molecules in the environment. And then, depending on the concentration of these molecules, they will engage in a particular kind of response. And these responses are all over the map. They include things like antibiotic production, the formation of biofilms, creating virulence, which is essentially creating a bacterial infection. So many of us, unfortunately, have had to experience bacterial infections. And these are due to the fact that certain microbial communities within our bodies are quorum sensing and expressing new genes. I'll note that bioluminescence was the pathway by which scientists discovered this mechanism of quorum sensing. And I'm honored to have a colleague at USC, Ken Nielsen, who was instrumental in figuring out the quorum sensing mechanism. And essentially they were seeing that these squids were bioluminescing, but it wasn't actually the squids, it was the bacteria that were living on the squids. One of the holy grails for those who study quorum sensing is with respect to bacterial infections and trying to see if we can come up with mechanisms to prevent bacterial infections and thereby reduce our reliance on antibiotics. So why have I emphasized this discussion of quorum sensing? It's because one of the key families of autoinducers or signaling molecules are these so-called acetylhomoserine lactone molecules, and this is a family also known as AHL, which are these signaling molecules. And they often react with other molecules in the environment to produce new molecules. And in a way, this is a form of degradation because the original molecule, which is shown by these triangle Bs, is specifically designed to bind to key receptors on the receiver end, if you will, of these bacteria, whereas the new products indicated by BEB, they actually do not necessarily bind. And so if all of these reactions are occurring in the environment, and they do, all of the AHL that's being produced by, say, the transmitter colony is not necessarily going to be received in time at the receiver colony. And so, as I said before, the products of these reactions may be uninteresting, and effectively, we have a packet loss, if you will. And so the key point here is that molecule degradation essentially puts a lifetime constraint on the molecule. And so if we look at our system where we have input signals that are being processed by these reactions, as well as unwanted reactions that are now being transmitted over the propagation channel, and then they are being received. All right. And so the key question we want to understand is, given that we know that molecule degradation can occur, we should take this into consideration as we schedule the reaction times and processing durations, if you will, at the transmitter, so that we can minimize end-to-end delay while also limiting the amount of energy that we use. So essentially, this boils down to us now having two constraints. We have this initial set of constraints. So if I have an arrival of a molecule here, I need to process it quickly before it degrades. On the other hand, I don't want to take too long to process it, because if I do, I'm not going to satisfy my delay constraints. This now means that I have a sort of valid region, which is the intersection of these two regions, that says this is the region in which I can effectively do my transmission over my propagation channel. And so this essentially converts our problem into having these two delays. Now, I need to talk a little bit about our energy function, which relates processing time to energy. And we make a number of key assumptions on this objective function, and that it's decreasing in the argument tau. So if I take longer time to process, then I'm going to use less energy, and it's strictly convex in this time tau. What's important about this problem is that the optimal algorithm does not actually need to know the actual form of the objective function. All it exploits is that these two assumptions that I've mentioned, decreasing an argument tau and being strictly convex. And as I said, experimental observations suggest that this very engineering approach to the design of the objective function is actually biologically consistent. So I'm not going to go too much into the details, but essentially we set up this optimization problem that has a series of constraints. There are so-called non-idling constraints. We don't want to waste any time durations by not actually transmitting signals. And then we have these so-called post-transmission delay constraints and pre-transmission delay constraints, which relate to the don't process too early and don't process too late constraints that I previously mentioned. So what can we say? We can say that we have an algorithm that's provably optimal. It's a moderate complexity strategy. The correctness of the algorithm is shown by the adaptation of majorization theory, and of course, direct exploitation of these two properties of the cost function that I mentioned. And it holds for all convex energy functions that have these desired properties. And so the ultimate solution is essentially to try to balance the transmission durations of all of the molecules that we're processing. And this is because we want to make our durations as long as possible, because that's going to minimize our energy use, but we still want to make sure we meet our final delay constraints. So in this first example, you see those X's, which are arrival times of our molecules, and we see the transmission durations that are designed for them. So molecule 1 is going to leave almost immediately and take up this amount of time. Molecule 2 is going to be later. This is 3 and this is 4. And in the first example, given our allocation of given our delay constraints and the arrival times, we see that we can design a strategy, or the output of the optimization algorithm is that everyone uses the same duration, and this is going to minimize our energy use. In the second example, we can still meet our delay constraints, but because the fourth molecule arrives too late, we do not have the capability of actually giving everyone an equal duration. So the first three molecules have an equal duration, which again is as long as possible to minimize the energy, whereas the fourth one, we use the time that is left over, if you will, in order to do the transmission. So this is a solution strategy that inherently puts a self-regulation mechanism onto our bacterial transmitting colony. All right, they are not transmitting as fast as possible and with as short durations as possible. We as engineers often seek these kinds of solutions as fast as possible, et cetera. But in the biological systems that we've studied, we've seen that there is, in fact, this natural self-regulation. And we've seen this in particular in our study of the activation of quorum sensing, where in our most recent work, which was published in iScience in April, we were able to show that natural systems do in fact engage in delayed gratification, because this delay is going to result in a healthier microbial community versus being greedy, which is in contrast to what we often see in purely engineered systems, where often greedy solutions are very close to optimal. So, as I said before, this particular problem was motivated by our interaction with our microbiology colleagues in trying to understand the systems that they were building. And so this is an example of a microfluidic system out of Ophelia Venturelli's group. And we realized that we also saw that there was sort of a saturation of the signals that were being generated by these bacterial communities. So we also took a look at the dual problem, which is minimizing delay subject to energy constraints, while also considering this molecule degradation. And this is a dual problem, all right? And so it's actually a straightforward adaptation of what we did before. And as before, we can design an algorithm that's provably optimal, that minimizes the completion time. We can provide necessary and sufficient conditions. And essentially here, you want to deplete your energy budget, but you want to make sure that your energy is optimally allocated within each allocation. And so this is a nice additional result. Now, our motivation in looking at this problem, and this first scheduling problem that we looked at, is a single hop problem, as seen in that MISTIC system, where I have one propagation channel between transmitter colony and receiver colony. But ultimately, we're very interested in these relay or multi-hop systems, which of course have been persistently studied in electrical engineering since the 1950s. And there's a ton of work in this space, and we ourselves, both from the biological and non-biological aspect, have actively studied this problem. But in trying to model the new microfluidic systems designed by the Venturelli group, we realized we had to make some different assumptions. So in terms of the scheduling work, let me just say that we are actively trying to look at this relay type system and multi-hop system. And we find that the way to adapt it is to think about multiple reactions. Our initial finding is rather significant, because before, in the single hop case, our optimal scheduling was completely independent of the particular energy cost function. I just needed convexity, but this is not true for the multi-hop case. So I actually need to know what that cost function is. Another challenge that we're actively overcoming at the moment is this prior work that I just showed you. We actually explicitly knew what all of those molecule arrival times are, and that's unlikely to be true in an actual system. So we're designing online mechanisms that allow us to do the scheduling in an informed manner. And of course, we want to do experimental validation, which turns out to be more challenging than one might expect. So let me talk a little bit about doing a communication modeling of these relay systems motivated by this original microfluidic device. It's called Fairy, out of the Venturelli group. And what was interesting and important here was to recognize that the arrivals at each of these cells, and each of these cells that you see here, each contain their own colony. So N0 is the transmitter colony, then I have N1 and N2 that are relay colonies, and then I have my receiver colony. And there are two mechanisms that we need to take into consideration. Importantly, arrivals at each of these wells are affected by its own past emissions. And so we have both external arrivals as well as self-feedback. And so the other thing that we need to take into consideration is a coupling between the colonies, because they actually compete for resources. And so the so-called, if I'm taking a amplify and forward as well as decode and forward model here, the gain of my amplify and forward is a function of the competition of resources between colonies. So I need to take the strain type, the number of active cells, as well as the range between these two. And so this is something that we have to integrate into the model that affects both how I scale my signal before I transmit it on, and whether my signal is significant enough in order to be transmitted on. And that's sort of the decode and forward aspect of things. And so in the end, for our first approach, we took a pure amplifier and forward scheme so that gamma factor is equal to 0. We assumed that the law of large numbers held, so we actually looked at a deterministic equation to govern our signals. And so what we had to compute here is essentially this channel matrix H, and that we assumed to be common for each one of the links that we were looking at. And so our end-to-end receive signal is a function of multiple applications of this channel matrix H, depending on the relay number or location, right? So the penultimate relay goes through only one channel, the one before that goes through 2 channels, et cetera, et cetera. And so you see here that in the end, we can create a description of our received signal at the receiver as a function of the signal sent out of the transmitting colony. And this is cleanly captured through a set of equations that capture the fact that not all signals are received, as well as the fact that we have this self-feedback. As I said before, we also had to worry about saturation. So what you see here, this N1, is the signal energy is a function of time that is being received. And we see that there's a saturation at some point. For each of the different relay systems, we see that the saturation occurs later and later, which makes sense because it takes more time to reach each of the relays. And so this particular saturation value was also taken into consideration when designing the model. So I'm going to close by noting that as we would expect, because of all of these sort of competing factors, competing for resources, self-feedback, not all signals leaving the relay in order to go to the next relay, there is actually an optimal number of relay systems given a particular formulation of the experimental system. And this actually is something that's quite important to our microbiology colleagues, because as I said, they are interested in trying to minimize reaction times between transmission and reception. And so we were able to show them that designing a system that had, 3 relays and therefore 4 hops was the way to minimize the reaction time. The first system that I showed you with the microfluidic multi-hop system, they've had some issues with reproducibility. And so now they're looking at a new microfluidic system as shown below, which is done with a very different design because bacteria are essentially put into these little pockets and then put into the cell. And so we're now working to modify our models to accurately reflect this system, which is slightly more robust with respect to reproducibility. So let me end by saying that I think in this area of biological communications and the modeling of biological communications, we have very much been taking the perspective of how can we take our engineering insights and use them to do this sort of modeling and design. But I think we're now at the point where actually some of our insights from this biological communications work can actually influence engineering systems. And as a point, when we first started working on these two-sided delay constraints, we felt they were only applicable to this biological problem that was given to us by our microbiology colleagues. But in fact, two-sided delay constraints are increasingly becoming a challenge in modern systems. So for example, if you're trying to coordinate action between a number of agents and information freshness is important, as in you're exchanging information, but if that information becomes stale, it's no longer relevant to the cooperative action you're engaging in, but you still want to meet delay constraints. This is an example of a two-sided delay constraint problem. Also in security systems where you have multiple sessions of communication and you're potentially using new keys for each communication session, that key can become stale and you do not want to use it multiple times. So you have to ensure that you're doing your transmission, your communication in a timely fashion before your session key no longer is relevant. And so I think this is an exciting point that we're at, where we can kind of go full circle with respect to engineering, enabling understanding of biology, design of biological systems, allowing us to design new engineering systems. So let me stop here and simply shortly say that we have this new bacterial signaling model. We're looking at bacterial colonies and multi-hop systems as schedulers. This invokes novel two-sided delay constraints, and we've been able to derive optimal scheduling algorithms for these systems. This original scheduling problem was influenced by our desire to handle these multi-hopped microfluidic systems, where in parallel, we're also designing models for the communication system. And as I said, things are applicable to other domains. Thank you very much.